Should be live now.
It's good.
Okay, perfect. Okay, so let's get started. Hello, everyone. Thank you all for joining us today. My name is Adriana Maldonado Chaparro. I'm a professor at Universidad de Rosario in Colombia, and I'll be your host for today's seminar. Before I announce our speaker for today's seminar, I would like to, thank Demian Farin for his talk on uncovering the hidden social life of birds. And I will also give a hint on our next speaker, Janet Mann. She'll be talking about social bonds, reproduction, and survival in wild bottle dolphins. And so please, I know some of you will have to run right after the talk, so just welcome to come to our next seminar next week. Just keep an eye for the email and the tweet that goes along with it. Just few reminders for our seminar today. Remember, this is a 45 to 60-minute talk, followed by anything in between 30 to 60-minute discussion, very lively with Liz, who agreed with us to join us and do this. Just please keep your mics muted throughout the talk, and then at the end for the discussion, just write or type down a question mark if you have a question, and if you're a student or a postdoc, type an S and we'll give you priority. If you are on YouTube, type in your question. Lauren kindly agreed to keep an eye on that, and he'll relay the questions to us. So it's my absolute pleasure to welcome Liz Lange. Liz got her PhD at Florida State University in 2019, right? Very recently. And then she continued as a postdoc in Susan Albert's lab at the University of Duke. And after that, she just recently moved as an assistant professor, and I will have to read this, Oswego, is that how you pronounce that? I hope so. State University of New York. So she just started building up her own lab, but she's been really successful. You can go and look at her website. Her lab employs a variety of techniques, including fieldwork, lab experiments, and theory. And her aim is to understand when and how individuals use information from their social environment to alter morphology, behavior, and life history traits. In addition to this, Liz and her team also seek to understand how the social environment affects survival and reproductive success. As you can see in one of the papers she sent us to read. So Liz's commitment to advancing the frontiers in animal behavior is demonstrated through her publication record, featuring contributions in journals such as Rugby, Neuroscience and Behavior, Science Advances Animal Behavior, Evolution, The American Naturalist, and many others. Her dedication to research extends beyond the laboratory, and she actively enjoys in mentorship, guiding the next generation of scientists. And she's also a fervent advocate of diversity, equity, and inclusion in STEM, and works very hard to create more inclusive and equitable environments. Also, she's a great communicator in science, and she's participated in various outreach programs, making science more accessible to the broader audience. So for today's talk, our title seminar is Setting the Foundations for Life, Long-Term Effects of Early Life Environments. Liz, thank you very much for accepting our invitation, and the mic is all yours.
Great. Thank you so much for the invitation and that very generous introduction. And thanks everyone for coming, taking time out of your busy schedules to talk, hear some of my research. So y'all can see my screen okay? Do that like awkward thing. Great, perfect. Great. So as Adriana said, I'm Liz Lange. I'm currently an assistant professor at SUNY Oswego in upstate New York. And today I'm going to talk about some of my postdoctoral and ongoing work on the Amboseli baboon research project, which I primarily did at Duke University. And so my work focuses on, there we go, understanding the factors important to differences in fitness, which is a key question in biology. So we know that developmental and environmental processes during early life can affect adult phenotypes, and that adult phenotypes can affect fitness, which is often measured by reproductive success or survival and lifespan. And of course, differences in the environment, whether that be abiotic, biotic, or social, can affect both early life and adult phenotypes, which ultimately affects fitness. Often studies, including many of my studies, focus on one part of this causal chain. So for example, determining how early life experiences affect adult phenotypes or how early life affects fitness. But in many systems, it's unclear how these processes are linked throughout the life course. So part of today's talk, I'm going to try to convince you that it's really important to understand how processes are linked throughout the life course. And so, for example, many studies in humans have shown that early life experiences are important. Adverse child life experiences like abuse, neglect, and household dysfunction can negatively affect developmental processes. And ultimately, this has been shown to lead to negative behaviors and health in adulthood, like increased rates of smoking, poor mental health, and ultimately early mortality. However, it's unclear how these early life experiences, in tandem with adult experiences, translate to differences in lifespan. And humans are not unique in the importance of early life experiences. So throughout taxa, you can probably pick your favorite taxa. Research has repeatedly demonstrated the importance of early life experiences on a multitude of adult phenotypes, including social behavior and fitness. In addition, we know that across taxa, including humans and other social mammals, positive social relationships are linked to longer lifespan. So understanding the links between early life adversity, adult social behavior, and health and survival is a really important question. However, a remaining question, particularly in social species, is to the extent to which early life effects on fitness-related outcomes are mediated by adult social behavior. And so what I mean by that is that when we see effects of early life on health and survival, to what extent are those effects driven by a link between early life environments and adult social behavior, which in turn affects health and survival? Or an alternative is that early life environments bypass the adult social behavior pathway and affect outcomes through some other route. And this is a really important question because, as I said, in humans and other mammals, individuals with stronger and more numerous social relationships have better health and survival. So it's really important to understand what is exactly affecting fitness. In addition, by assessing the time points in the life course to which it has the strongest effects on health and survival will allow us to key in on potential mitigative solutions to poor early life experiences. And so when we're thinking about, social behavior evolution and the relationships between early life, adult social relationships, and survival, we really need to make progress on several key questions. So the first key question that we need to understand for social behavior or many other phenotypes is, what is the genetic basis of social behavior? And this is important so that we can understand if the trait has a genetic component and thus can adaptively evolve via natural selection. We also need to understand the environmental sources of variance in social behavior so we can determine the developmental mechanisms that are important to this fitness-related trait and assess the potential for phenotypic plasticity, which is really important in the face of human-mediated impacts, including climate change. And then finally, we need to understand how social behavior contributes to survival and reproductive success in adulthood and throughout life. So today I'm going to tell you about some of the progress that I've made on these questions with my work in collaboration with the Amboseli baboon research project. Specifically, I examined first how social behavior develops by assessing the genetic and environmental determinants of juvenile social behavior by examining the age at which an individual first grooms a social partner, which is an important affiliative behavior in baboons. Second, I'm going to tell you about some of my work linking the effects of early life adversity, adult grooming patterns, and fitness. And then finally, I'm going to talk about if and how animals can be resilient in the face of poor early life environments by assessing if positive social relationships can mitigate or reduce the negative effects of early life adversity on survival. And so by doing so, I hope I'll be able to convince you that by considering multiple time points throughout the life course, linked together is a really important and essential thing to do to understand how social behavior might evolve. So first, I'm going to tell you about a study where we assess the genetic and environmental determinants of age at first grooming. And so we know from many different taxa that positive social environments early in life are linked to better social relationships and survival in adulthood. But despite the well-described role social interactions play across the life course, less is known exactly how affiliative social bonds develop and what environmental and genetic factors particularly predict variation. And so in particular, understanding if the development of social behavior is heritable is an important gap in the literature. And so it's important to assess heritability because it describes the amount of phenotypic variation that arises as a result of additive genetic variation. And when I say additive genetic variation, I mean the variation that describes parent-offspring resemblance. And so narrow sense heritability is calculated via the equation here, where additive genetic variation is divided by the phenotypic variation. And it's key to assess the heritability of social behavior because a heritable component is necessary for evolution by natural selection. And so in collaboration with an amazing undergraduate at Duke, Madison Griffin, who's now a master's student at the Virginia Institute of Marine Science, we assessed the genetic environmental predictors of age at first grooming. Grooming is an affiliative social behavior in many primates, including baboons, where individuals systematically pick through the fur of another organism. And so today I'm going to tell you about some of our work assessing how, if and how sexes differ in their first grooming behavior, what the environmental predictors of age at first grooming are, and then if there is a heritable component to age at first grooming, thus allowing it to evolve adaptively. And so we asked these questions in a wild primate population, the baboons of the Amboseli Basin in southern Kenya. This system is in right outside of the Amboseli National Park and right outside of Mount Kilimanjaro on the border of Kenya and Tanzania. The Amboseli Baboon Research Project was started by Jean Altman and is currently run by 3 project leaders, Susan Alberts, Jenny Tung, and Beth Archie. All of the data are collected by the field team with help and support staff pictured here, and I wanted to take a minute to truly acknowledge Kinua, Raphael, Longita, and Sarah, who without which there would be no Amboseli Baboon Research Project because they collect a majority of the data. We've been collecting behavioral, ecological, and demographic data for over 5 decades. And the Amboseli baboons are highly social primates that live in social groups of 20 to 150 individuals of mixed sex, and making them an ideal system to ask questions about the links between early adversity, social behavior, and fitness. Previous work has shown that stronger adult grooming relationships are linked to longer lifespans in both males and female baboons, but it's less well known as to how these grooming relationships develop. And so to make progress on this important question, we assessed age at first grooming, which we defined as the age at which a subject was observed to coordinate both hands to systematically pick through the fur of a second animal, which is grooming. And again, grooming is primarily affiliative social behavior in the baboons, so it's an important behavior to understand. So the first question we asked is whether males and females differ in their first grooming behavior. And we did this by using a general linear mixed model, and we found that males and females do differ. So here I'm showing you the age at first groom on the x-axis and the probability of grooming on the y. Females are in red, males are in blue, and we see that female baboons on average have a median age at first groom about 2 months earlier than male baboons. And this might pinpoint and describe sex-specific differences that occur throughout life. So female grooming relationships are key to female fitness, and while male grooming relationships are also related to fitness, they do not groom quite as often as females. And so this might suggest that these sex-specific differences that we see in adult baboons start really young. So second, we wanted to ask if they differ in their first grooming behavior in terms of which partner they groom first. And so here I know is a busy slide. I'm happy to talk more about this in the question and answer. But here I'm showing you the subject sex on the X, so female subjects and male subjects. And I just want to show you that the warm colors represent a female first grooming partner, and the blue colors represent, or cool colors represent, a male first grooming partner. And so what we found is about 70% of both female and male subjects groom a female first, and this did not differ between the sexes. And in addition, if we can, on this darkest warm color, that is the proportion of individuals who groom their mother first. And so mothers were the first grooming partner more often than random chance. However, this did not differ between males and females. So while males and females differ in the age at which they first groom an individual, they don't differ in the partner that they are grooming. And the fact that mothers were first grooming partners is probably not surprising. Young baboons do spend a majority of their time in close proximity to their mother. So second, we asked about the environmental predictors of age at first grooming behavior. And so to do this, we used a Cox proportional hazards AICC based model selection because we did not have specific hypotheses about a multitude of different environmental factors that could be important in predicting age at first grooming. So the environmental factors that we assessed were two, which were features of the social environment. So whether or not an individual had maternal siblings and how large the social group size was. Two were features of the physical environment. So whether an individual experienced drought in the first year, and then what season they were born in, the wet or dry, because these physical environments may have something to do with resources available to the developing baboons. And then four had to do with maternal environment, because we know mothers are really important in the development of social mammals, especially primates. And so first we examined maternal experience, so whether the mother had a previous offspring or if this was her first offspring. We assessed two components of maternal rank, proportional rank and alpha rank, where alpha rank is whether or not the mother was the top ranking female in the group. And then finally, we assessed maternal social isolation in the first 2 years with the hypothesis that mothers who are more socially isolated may be affecting the social partners available to their offspring. And so we considered models with delta AICCs less than 2 from the best fitting model as the top models. And we ran separate models for males and females because previous preliminary results had shown that they are different in their age at first grooming, as well as the fact that when we included sex in these Cox proportional hazard models, they violated a key assumption of proportional hazards. We also included observer sampling effort as a covariate because the rate at which baboons are observed can and does affect the age at which individuals are first seen grooming. So the strongest and most consistent predictor of age at first grooming was group size. And all models best fitting for both sexes included a term of group size as well as observer sampling effort. So here I'm showing you time to event curves for age at first grooming on the x-axis and the probability grooming over the y for females and males. Group size, the smallest group sizes are in red, the largest are in gray, and the intermediate group sizes are in blue. And for both females and males, we see that those in the largest groups groom later than those in the smallest groups. For females, individuals in the smallest groups groomed at about 10 months, whereas those in the largest groups groomed 2 months later. For males, this difference was even more dramatic, where individuals in the smallest groups groomed at about 10.7 months, and those in the largest groups groomed 4 months later at 14.5 months. And so we hypothesized that one factor that could be coming into play to explain these results is that males and females in many social mammals differ in the amount of play that they are performing. And in larger groups, there are more closely in age peers for individual developing baboons to play with. And so the lack of age at first grooming in males and females in larger groups, or the delayed grooming in males and females in larger groups, might be a factor of other social behaviors that these individuals are performing. In addition, in some species, larger groups have generally less affiliative social behavior and more aggressive social behavior. So that might also explain these results. And of course, future work should really assess what is driving this interesting relationship. So for females, I showed you that social group size plays a role in age of first grooming. We also found a weak effect of drought in the first year, whereby late groomers who groomed after the median age at first grooming were more likely to groom earlier if they experienced drought. I'm happy to talk more about that later. For males, we again saw this effect of social group size, but there is also an effect of both maternal rank parameters, such that males with alpha mothers were more likely to achieve first grooming at any age compared to males with non-alpha mothers. But when controlling for alpha status, males with high-ranking mothers were less likely to achieve first grooming at any age compared to males with low-ranking mothers. And so this is an interesting result where we see this alpha effect, and this has been described for other types of phenotypes in the baboons, such as glucocorticoid levels. So asking about alpha effects in baboons and other Cercopithecine primates with dominant systems represents a really interesting avenue of future work. So finally, we wanted to understand if there was a heritable genetic component to age at first grooming. And so to do this, we use an approach known as the animal model, which is used extensively in livestock breeding, but also in evolutionary biology. So the animal model is a mixed effects linear model, and the response variable is the trait of interest. In this case, we examine the age at first grooming. In the animal model, you can include fixed effects, which are environmental variables shown to affect variance in the trait of interest. So in this case, we included sex, group size, and observer effort. And then in the animal model, to assess a genetic component, you include a random effect of a pairwise genetic similarity matrix between each pair of individuals. And this is calculated from a pedigree. And so to get the heritability of a trait, you assess the proportion of variance in the trait explained by genetic similarity. So if we consider this example of two individuals with a phenotype of interest, let's say age at first grooming, controlling for environmental effects, if there's a heritable component, we might see that unrelated individuals are not correlated in their phenotypes, but half siblings do show a signal of having similar phenotypes. And that would be even stronger for individuals who share more genetic components, such as full siblings. If there's not a heritable component to the phenotype of interest, then we wouldn't expect any relationship between genetic similarity and phenotypic similarity. And so to assign relatedness, we constructed a pedigree. Our pedigree example is shown on the right here. And so to construct the pedigree, we use paternity assignments from fecal-derived DNA constructed over about 15 years. Our pedigree includes approximately 600 individuals for whom fathers have been assigned and 1,500 individuals total. And this makes this the largest pedigree for any wild primate in the world and probably the second or third largest for any wild mammal. So this is a really powerful tool to ask questions about the genetic basis of behavior. For the age at first grooming analysis, we included the 731 subjects that I've been focusing on before. And for these 731 subjects, the pedigree necessary to assess relatedness was about 1,300 individuals, 871 had known mothers, and 531 had known fathers. And so before I show the result, I wanted to build the animal model components one by one. I just told you about some of the fixed effects, the environmental sources of variance in the behavior, so sex, group size, and observer sampling effort. And to those fixed effects, we include a random effect of maternal identity, because each mother can contribute multiple offspring to the data set, and the mothers may influence their behavior through maternal effects. So for instance, the mom's behavior might influence the age of first grooming of their offspring. We also included random effects of social group and birth year to control for similarities related to these important environments. And then we add a random effect of genetic relatedness, that's this GI term here, and an error term or residual variance term. And so we call this the heritability model. And we compared the heritability model to 3 other models to assess using DIC values if it was the best fitting model. So we also compared this to a maternal effects only model, which included the fixed effects, but then only a random effect of mother. We also compared it to an environmental effects only model, which included fixed effects and the other random effects, but no G term. And then finally, we compared all models to a model with all of the random effects, but no fixed effects, because fixed effects in the animal model can reduce residual variance and thus alter heritability estimates. So we compared these different models to understand which fit best. And so first I'm going to show you the intercept only model, which did not have any fixed effects. And this model, I'll tell you outright, did not fit as well as the models that included fixed effects when we compared DIC or deviance information criteria, which you can think of as AIC values. In the intercept only model, about 4.5 of the variance in age at first grooming was explained by an additive genetic effect shown by this yellow bar. When we assessed the other models, we found that the model that included the additive genetic variance term, the heritability model, fit best. And so this best fitting model included the additive genetic component. And from our estimates, we found that the heritability was weak and equal to about 0.04. But it had credible intervals that did not overlap 0, suggesting that this was a weak but significantly different from 0 heritability estimate. We do have a lot of variance that's unexplained, and so that's an interesting avenue that I'm happy to talk more about in the question and answer as well. As a whole, these suggest that we have a significant but small heritability, such that natural selection can affect this trait, but environmental factors are also really important. All right, so to sum up this portion of the talk, I've showed you that males and females differ in their age at first grooming, with females grooming earlier than males, but they do not differ in their first grooming partner, where both tend to groom females, particularly mothers. I showed you that social group size was an important environmental predictor of age at first grooming for both sexes, and then drought affected female age at first grooming, where maternal rank affected male age at first grooming. And then finally, I showed you that age at first grooming is weakly heritable, suggesting that it can evolve. And so age at first grooming is a really important social developmental milestone in the baboons, but it doesn't tell you, we don't know a lot about how it's linked to adult behavior. So as a preliminary analysis of this, we decided to link the development of mother-offspring bonds throughout the baboon's early life. And so we chose to focus on maternal offspring bonds because mothers are particularly important relationship to mammals, or especially early in life. So, for example, orphan elephants who lose their mother have reduced affiliative behaviors with adults, as well as shorter lives. And in the system I work on, losing the mother, who is a critical social partner during the juvenile period, even after weaning, can have lifelong consequences in terms of reduced lifespan and reduced social bonds. And so we asked how mother-offspring grooming equivalence changes with age. And so equivalence you can think of as the proportion of grooms a mother gives the offspring over the total number of mother-offspring groomings. So we're trying to get at, is the grooming relationship equal or driven by one particular social partner? And so here we use the binomial generalized linear mix model with the number of mother-offspring grooming as a weight. And we analyzed 2 time points separately. So our first time point, 0 to 54 months, represented a time period where both males and females were immature, but females reach menarche before males attain rank and have testicular development. So in this second time point, females were mature, but males were not. And so here I'm showing you the offspring age in months and the proportion of time a mother initiated. Perfect equivalence would be 0.5 here in the dotted line. Sons are in blue and daughters are in red. So first, we can zoom in on daughters. Daughters reach grooming equivalence with moms first at about 38 months. They then overshoot and come back to about perfect equivalence in their early to late adulthood. Male offspring reach grooming equivalence later at about 58 months and then maintain about equivalence throughout the time that they're in the group with their mother. So next, we wanted to understand who was driving this relationship. Was it the offspring changing their grooming relationships with the mother, or was it the mother changing their grooming relationships with the offspring, or a combination? And so here I'm showing you again offspring age in months, and then the number of times the offspring initiated a grooming event. And this figure is showing that female offspring tend to change their grooming patterns more across their lifespan than male offspring. And if we key in on the number of times the mother initiated a grooming event with their offspring, especially during the time in which both males and females are immature, we do not see a difference between male and female offspring being groomed by their mother. And so this suggests that sex-specific changes in grooming equivalents, at least early in the offspring's life, are largely driven by daughters grooming mothers more and not by mothers changing their behavior based on the sex of their offspring. So this pinpoints the really important relationship that mothers have in the development of their offspring and how grooming relationships might develop. And future work, I would love to assess how different grooming relationships develop as well. So this first half of the talk, I've told you a little bit about the genetic and environmental determinants of this really important early life phenotype, age at first grooming. And in the second part of the talk, I want to tell you about linking early life to adult phenotypes and fitness. So I'm going to tell you about the effects of early life adversity in adult grooming and fitness, as well as the potential for mitigation of negative early life adversity effects on survival by positive adult social relationships. And so as a reminder, we know across species, including humans, that early life experiences are important, and they have lasting effects on social behavior and fitness. And remaining question here, though, is the extent to which early life effects are mediated by adult social behavior. So when we see effects of early life environment on adult social behavior and effects of adult social behavior on survival, how much of this is driven from the pathway that links early life through adult social behavior to health and survival, and how much of this relationship of early life on survival bypasses the adult social behavior phenotype. And so in the baboons, we have evidence for all of these links. So we know that early life adversity is linked to decreased lifespan. We know that early life adversity is linked to weaker social bonds. And finally, we know that adults who have weaker social bonds have poorer survival. And so the obvious hypothesis here is that social bonds are a mediator, but this is yet to be explicitly tested. And so to measure this mediation effect of adult social behavior, we focus on adult female survival. And this is because males disperse when they reach sexual maturity. So assessing male fitness is considerably more difficult. We ran separate models for the mediating and moderating effects of female-female bonds and female-male bonds because they differ in their function and how strongly they affect survival. Today, for the purposes of this talk, I'm going to focus only on female-female social bonds, but the female-male social bond results were similar. So from the Amboseli Baboon Research Project, we compiled the data set of 199 females, and from these females, we have 1,849 years of life for social bond strength and 74 deaths. And again, we're only modeling females because males disperse from their natal groups, which makes estimating survival difficult. And so to measure early life environments, we measured the cumulative early adversity, which is the sum of 6 sources of early adversity. So 2 of these sources have to do with resources, so whether an individual experienced drought in their first year of life and whether they were born to a large group size. 2 have to do with the maternal social environment, so if an individual's mother was low maternal dominance rank or was socially isolated herself. And 2 have to do with family structure. So if they lost their mother before the age of 4 years, or if they had a close in age younger sibling who would deter some of the mother attention. And so to calculate the cumulative adversity index, we assessed which of these adversities an individual experienced, and then we added them up. To measure adult social behavior, we focused on grooming because it's the primarily affiliated behavior in primates, and it's known to be central for establishing and maintaining social bonds. So we used the dyadic sociality index, which measures the social bond strength of the top 3 social partners based on grooming. The lower values of the dyadic sociality index mean weaker social bonds, and higher values indicate stronger social bonds. So we first asked, do the social bonds in adulthood mediate the effects of early life adversity on survival? So does early life adversities affect adult traits, social bonds, which in turn affect fitness, or do early life adversities bypass this pathway that includes social bonds? And so to test this hypothesis, we used mediation analysis with joint modeling, This is a causal inference approach that was developed by two statisticians pictured here, Dr. Fan Li and Dr. Shushi Zeng. And it allows us to take into account the longitudinal nature of social bonds and examine if they are this black box mechanism that links early adversity and survival. This modeling framework allows us to test if social bonds are part of a causal chain that links early life adversity and survival. So in other words, when I say a mediating effect, I mean that part of the reason that early life adversity affects survival is because it alters social bonds. Mediation analysis also allows us to estimate parts of this framework. So for example, we can determine the effects of cumulative adversity on social bonds, shown in orange here. We can also determine the effects of social bond strength on survival independent of an individual's early life experience, which makes this approach really powerful. We also can estimate the total effect of cumulative adversity on lifespan, which has been estimated previously, but this modeling approach allows us to break down the total effect into two parts. First, in green, what we call the direct effect, which is the direct effect of early life adversity on survival independent of changes in social bond strength. And then secondly, the indirect or mediation effect, which tells us how much of the effect of early life adversity on survival is due to early life adversity changing social bonds, which in turn alters survival. So when we look at female-female bond strength as a mediator, I'm going to build in the effects on this diagram. So for each additional source of early adversity a female experiences during the juvenile period, her lifespan is decreased by 1.6 years. Of this 1.6 years, 90% of this total effect, or about 1.4 years, is explained by the direct effect of cumulative adversity on survival outside of a pathway that includes social bonds. Therefore, lives of females who experience 4 sources of early adversity are predicted to be over 6 years shorter than those who experience none. And of those 6 years, 5.5 would be explained by the effects of early adversity on survival, independent of a pathway that includes social bonds. However, we do see an effect of early adversity on female-female bond strength. So individuals who experience more for each additional source of early adversity experienced, female-female bonds are decreased by about 0.09 DSI units, which is about 0.15 standard deviations. For every 1 unit increase in female-female bond strength, we see an increase of 2.2 years in lifespan. And we do see a mediation effect such that 2 months, which means that of the 1.6 years of the effect of cumulative adversity on survival, 2 months of that effect can be explained by the relationship between cumulative adversity, female-female bond strength, and survival. However, this mediation effect is really weak and only explains about 10% of the effects of early adversity on survival, suggesting that cumulative adversity and female-female bond strength are largely independent in their effects on survival. And early female-female social bonds in adulthood are not a strong link between these phenotypes. So I've just showed you that there's only weak mediation of early life environments via adult social behavior on health and survival. But an alternative hypothesis is that social bonds may play a moderating role. And so here we are asking if adults with stronger social ties despite early adversity able to reduce the negative effects of early life adversity on survival. That is, rather than linking early life environments and health and survival, social bonds would interact with early life environments such that individuals with stronger or social bonds are able to reduce the negative effects of early life adversity on survival. And so here we asked if social bonds influence the strength of the relationship between cumulative adversity and survival using a moderation analysis. And we predicted that social bonds would have a larger effect on survival when females experienced early adversity, and adult females who experienced early adversity but maintained stronger social bonds would have higher survival than adults who experienced early adversity but have weak social bonds. So to show you the results, I'm going to show you each of the 6 sources of adversity. So drought or rain in the first year, a close in age sibling, group density, group size, maternal loss, low maternal social connectedness, maternal rank, and then that cumulative early adversity index of 0 to 6 that we've been talking about throughout the talk. And the y-axis is the change in the interactive effect which means that positive numbers here would suggest that positive social bonds with females amplify the negative effects of early adversity, whereas negative interactive effects means that stronger social bonds with females in adulthood reduce the negative effects of early life adversity on survival. So social bonds are bad, social bonds are good. And for most of our sources of adversity, including cumulative adversity, there are no interactive effects of the social bonds with females and early life adversity. But for the strongest individual source of early adversity in our population, maternal loss, we find that individuals who lose their mother but are able to have stronger social bonds with females are able to reduce some of the negative effects of maternal loss. And this is really cool, and it makes sense because maternal loss, when you lose your mother early in life, much of what you're losing are those social connections. So mothers are an extremely important social partner, and individuals with a living mother, they are often their strongest or among their strongest social connections. So when you lose your mother early in life, you're losing that ability to form strong social bonds with your matriline, including your mother. And so to visualize this result, if we look at the social bonds with females, where on the x-axis, I'm going to show you individuals who did not experience maternal loss and those who did. And on the y, I'm going to show you the hazard ratio, where higher numbers mean lower survival and lower numbers mean higher survival. When individuals do not experience maternal loss, those who have stronger social bonds to other females are only about 10% more likely to survive than females with weak social bonds. But we do see that the slopes are different when we connect these to maternal loss, such that individuals who experience maternal loss but have strong social bonds are 16% more likely to experience, or less likely to experience mortality than those who have weak social bonds. And so there is this interactive effect between maternal loss and social bonds with females on survival, such that social bonds reduce the negative effects of early life adversity on survival. This means that if you do experience maternal loss and you're a baboon, you're not doomed, but it's really important that you make these strong social connections in adulthood. Okay, so for this part of the talk, I've showed you that early life adversity and adult social bonds both affect fitness. But more importantly, that social bonds with females only weakly mediate the link between early adversity and reduced lifespan. And that independent effects of early life adversity and social bonds on survival is the most supported hypothesis. I also showed you that social bonds in adulthood can reduce the effects of maternal loss on survival, again, suggesting that if you lose your mother early in life, forming strong social bonds is even more important. And as a whole, these results suggest that by considering the entire pathway of traits, we can gain insights that were not obvious from focusing on parts of the pathway. So for example, in previous work, we saw strong effects of early life adversity on adult social bonds and social bonds on survival, but making it, we hypothesized outright that there'd be this strong mediation effect, but that's not what we see. Instead, we see largely independent effects of early life adversity and reduced social bonds, which suggests that mitigating effects can pinpoint both early adversity and adult social bonds if we're trying to increase the survival of individuals. And so to sum up, I started the talk by formulating these 3 general questions, and I want to sum up what progress we've made so far, although there's a lot more progress to be made. So I've showed you the genetic basis of social behavior, whereby age at first grooming is weakly heritable. And we also know from previous work that adult grooming patterns are also heritable, more strongly heritable here. And that's a paper by Emily McLean et al. In Evolution. But taken together, it suggests that adaptive evolution can shape social behavior and is adding to a growing literature showing this important trait that's linked to fitness can evolve adaptively. I also touched on the environmental sources of variance in social behavior. So group size is a really important trait or environment for determining age at first grooming. And early life adversity as a whole decreases adult social bond strength. And then finally, the fitness consequences of social behavior is an open question for the development of grooming behavior and something that I think is really important to assess. But we did see that females with stronger social bonds live longer lives. And so as a whole, my work suggests that, social behavior is this key component of an individual's lifespan, and that by studying it throughout the life course, we can gain insights that were not immediately obvious from studying just time points in time. And so with that, I'd like to thank the organizers for inviting me to give this seminar. I'd like to thank you all for listening. And I'd like to make sure that I acknowledge the Amboseli Baboon Research Project, particularly our data managers, Jake Gordon and Nikki Learn, as well as the observers, Rafael, Sarah, Longita, and Kinua, without whom this project would not be as successful as it is. I'd also like to acknowledge the Alberts Lab, as well as the collaborators who are specifically involved in these projects I talked about today, the funding sources. And if you're listening to this on YouTube later, my contact information is there as well. So with that, I am happy to take any questions.
Thank you very much, Liz, for a great talk. I think we all enjoyed it very much. So this is the time for questions. And just as a reminder, please type in a question mark. If you have a question in the chat, do it to the everyone list. If you are a student or a postdoc, please type in S and we'll give you priority. And for those watching us on the YouTube, please type in your questions and we'll make sure to bring them into the seminar live. So our first question comes from Eduardo.
Thank you, Adriana. Thank you, Liz. Fantastic talk. So much I'm learning and so many details, therefore so many questions. A general comment I have is that I really appreciate the attention to some specifics of the methods that you have followed in the analysis of the data. And we seldom, I think, I include myself, we seldom attend talks or read papers where we consider sampling effort as an important factor. And I'm delighted that you mentioned it, even if it leaves me with an awful lot of doubts. But I'd rather have that than be wondered, what's the sampling effort? But I didn't quite follow because you brought it up. I'd like to understand, isn't this a huge potential complication for everything? And then in the conclusions that you gave us, the take-home messages, you did not include it. But can you explain me, please don't overestimate how much I understand all this analysis. So really, maybe that I'm just not getting it. So when you were saying that observer sampling effort is one of the strongest predictors, can you elaborate on how complicated that could be for everything else?
Yeah, of course. So you're talking about the age of first grooming analysis, right? Oh, you're muted, Eduardo.
I mean, that's the one for which you bring it up, and I appreciate it. I think that all of us need to much more look at sampling effort and variation, not to mention variation about observers, right? We talk as if they were clones in their ability to collect our data, but that's something else. But yes, you bring it up first for age at first grooming. Yeah,
so I mean, not surprisingly, groups who have higher observer sampling effort, individuals are observed to be grooming for earlier in those groups, right? And so we like to think of the age of first grooming measure as the age of first observed grooming, right? And that is clearly affected by the sampling effort. We do try to limit biases in sampling effort by using what we call representative interactive sampling, which means that the observers go through a randomized list of individuals and they do focal samples on those in each group. However, because there's, less individuals in smaller groups, they're going to hit those individuals more than in larger groups. And so that's where we really get a difference in the number of observations in the different groups in this case. So I think, sampling effort is something that we all need to keep in mind, especially when we're thinking about these age important milestones. But we also aim to, try to control for those statistically. Yeah. And with our methods.
Thanks. I need to, again, I'm slow to this.
Well, we can chat more about it.
Yeah, I need to keep processing, but thank you so much.
Thank you, Eduardo. Next in line is Phoebe.
Hi, Liz. Great talk. The maternal loss interaction data was so cool. I have a question about the first data you presented.
Phoebe, can you introduce yourself? I forgot to do it myself. I'm sorry to everyone. I'm Eduardo Fernández Duque from Yale, but we continue building our community. If you can just say where you're joining us from and your institution.
Sure. I'm Phoebe Edwards. I'm a postdoc at University of Toronto. So my question was in the first data you showed, I'm sorry if I missed this, can you reiterate why age at first grooming was such an important factor to look at versus something like frequency of grooming in the juvenile period or duration of grooming or factors like that?
Yeah, thanks, Phoebe. I think all of the factors that you mentioned are things that are really important to consider, right? The development of social behavior is not just the age at first, which, the age at which you first do this, but also how much you're doing it over the grooming, over the developmental period, how that might change with time. And so we chose age at first grooming as a first step. And then we expanded that to look at the grooming equivalents with the mothers. But I'd really like to then assess, how grooming behavior changes over the juvenile period, not just with the mothers, but measuring something like, the dyadic sociality index or something like that. So yeah, does that answer your question, Phoebe?
Thank you, Phoebe. Okay, so yeah, just a reminder, please state your affiliation, name, and where you're coming from, so we build a community around here. Our next question comes from Eugenia.
Yes, hello. I work in Rome, in Italy, and I work at the local health unit at the veterinary service at the dog shelter. Maybe I missed it, but first of all, thank you for my old lab were the primates. I took my PhD on primates. Then thank you. Maybe I missed it, but what are the ultimate causes? Because the young females began 2 months earlier than the males. Maybe I missed it.
No, I think that's a really great question. So are you asking about how that's related to fitness in future adult phenotypes? Okay, yeah, so we haven't assessed that yet, but that's, next on my list of questions, right? I think linking, as Phoebe said, like early or age at first grooming to subsequent grooming development, and then also adult, social behavior is a key question, as well as asking how, are these early groomers also, having any fitness related consequences, since we do know that adult social behavior is so tightly linked to fitness. So yeah, that's a next step project.
Okay, thank you.
Thank you.
Thank you, Eugenia. Next question comes from Juliane. Hopefully I pronounced
Hello. Yeah. Hello, I'm Juliana Dam. I'm currently doing my doctor at the Instituto de Neuroetología in Mexico, in Jalapa. I'm working on social coping and physiological stress. I was wondering in the first part of your talk about how you disentangle social learning and heritability, because I have the feeling that, especially in a primate that is very maternally bonded and doesn't really, if I'm correct, I work with neotropical primates, so but I have the feeling they don't really go outside of their little maternal bubble a lot. So I was wondering how you disentangle the social learning aspect from heritability. And the other thing that I would want to know is how you mentioned it quickly, that you can elaborate more on trot and how it affects the females and the early grooming period. So if you could elaborate on this a bit more, sorry.
Yeah, no, of course. Thanks for those questions. So to answer your first question about disentangling, social learning potentially from the mother or other social, partners in the group. That's why we also included those random effects of maternal ID as well as social group in the analysis. However, that's a really great question, in understanding, how particular mothers might influence the development of these offspring is a really interesting question. I will say that by the time they're doing their, performing their first grooming, they're not as tightly linked to their mother. It's, common that they'll be going, they are tightly linked to their mother, but they're also foraging away from their mother, foraying away from their mother, not foraging. But yeah, so we did try to control for that statistically. And then in terms of your second question about the effects of drought, they were quite weak, but so here I'm showing you females who experienced drought in orange and those who did not. So there weren't any differences in up until the median age of first grooming, but the hazard ratio confidence intervals didn't overlap. One, suggesting that individuals who are groom after the median age, or what we like to call late groomers, those who experience drought are grooming earlier than those who didn't experience drought. But this is a really interesting pattern that we haven't quite wrapped our heads around, explanatory factors that we could test explicitly. Yeah.
Okay. Thank you.
Thank you.
Thank you, Julian. Next in line, Anthony.
Hi, my name is Tony DeFury from the University of Texas at Austin. Liz, thank you for a really interesting talk. I have to say, as somebody else who studies neotropical primates, I get baboon envy with the sample sizes that you guys have and the amount of rich behavioral data that you've collected over 50 years. So it's great to see it all put together. I had a question for you, and also I'll second what Eduardo said. I really like the mediation analysis and your detail on that, right? Getting at the causal links in that chain from early adversity to adult survival. I wanted to ask about the adversity index and maybe ask you to talk a little bit more about that. If I understood correctly, it's a sum that's going to go from 0 to 6, right? So the value from that's going to be 0 to 6, and you're combining things that have a lot of, you're combining a bunch of different things into that adversity score. And so maybe if you could just talk a little bit more about what went into the choice of those variables to include in the index, and then also comment on, the results of the model then suggest that a change of 1 unit in that adversity index results in a change in X units of survivorship, or X degree of strength in the X strength of the social bond. And so can you put a more biological interpretation onto that adversity score, which is an index that encapsulates a bunch of different things?
Yeah, of course. Okay, so here is the adversity slide that I showed y'all before. And so these 6 sources of early adversity were based on the Tung Archie paper in 2016, who first established that these early life adversities can be linked to decreased adult survival. And so to be consistent and be able to relate to other research done in the lab and with the project, we chose to, maintain the same sources of adversity. Did that answer your question on that, Tony?
Yeah. So it's a score though that's going to range. Each of these gets
Yes.
a 0 or a 1, right? And so
0, yes.
Right. So you're comparing or treating in the index. And I don't know, you know the statistics associated with this probably a lot better than me, but you're treating in the index a value of 1 as having putting equivalent weight on drought in the first year of life versus being in a large versus a small group at birth,
Yes.
right? And so you're kind of combining a lot of different things into an index and giving them similar weight. Is there? So that's, I guess, what I wanted to see.
Oh, yeah. Okay, yeah.
Correctly.
No, thanks for clarifying. So
Yeah.
yes, that's what we did. So each of these is based on, presence or absence of these, based on quartile cutoffs, or, in terms of maternal loss, it's you either lose your mother or you don't. And the cumulative adversity is literally the sum of, did you experience drought 1, large group size 0, et cetera, et cetera. So if you only experience one of these, your score would be 1. In practice, it can go up to 6 sources, but we don't have any individuals who experience more than 4 or 5. And so it does lump a lot of different sources together. And part of the reason we decided to assess this is because the human literature, which we were modeling after when we initially started this, and by we, I mean, Jenny, Beth, Susan, and Jean, often uses this adversity index where they sum all the different adversities together. Now, there are obvious limitations to that. And so in addition to assessing this cumulative early adversity score, we also ran models with each of the 6 adversities independently to assess how those affected in this mediating pathway. And we found that drought in the first year of life and maternal loss were the 2 that came out as the strongest sources. But understanding exactly, this cumulative adversity index and whether these sources are summative or interactive, I think is a really important question that some people are just starting to grapple with. And I did a little bit of work, which I didn't talk about that today, on that. Unfortunately, even with our huge sample size, we don't quite have enough power to assess the interactive effects of these adversities. But it's something that I think is a really interesting and important question. Yeah. So that's my piece on the cumulative adversity index. I think by assessing these individual sources as well as the adversity index, we can get a better idea than just using the summative index. So that's that. And then what was your second question? Sorry, I forgot it already.
I don't know if I had a second question. Oh no, it was about a change in one unit in your model, a change of 1 unit in the adversity score
Okay.
is associated with some kind of fitness effect or some kind of impact on the strength of the social bond, right?
Right.
And so,
0.9 DSI unit or 0.9 is not really informative, right? I mean, it is and it isn't. But when we think about that number, we can also think about it in standard deviations. And so the standard deviation of social bond strength with females is about 0.59. So 0.09 DSI units is about 0.15 standard deviations. So that's one way we think about, that in a more realistic way.
I guess the point, I'm sorry.
So then 2 standard deviations increase in female-female bond strength increases survival. So the effects of early adversity on female-female bond strength are much weaker than the effects of female-female bond strength on survival.
Right. I guess my point was that you could have a change of 1 in the adversity score by either losing your mom or experiencing a year of drought or having a competing sibling for your mom's attention, right? And so it seems I think it would be worthwhile. I think it would be worthwhile to present as you do the presentation on kind of the cumulative index, including those other analyses that I think you said you did and referred to a little bit, looking at those scores, those adversity measures independently, because some of them are, and I think you acknowledge this, drought and maternal loss are really the ones that are driving the effect.
Yeah, and so for drought and maternal loss, the effect on the female-female bond strength is about half of a standard deviation, so it's much greater than this cumulative index. Yeah, it's just for timing, we have to cut something, and so yeah.
Thank you. I just wanted to make sure that I understood. Yeah.
No, you totally understand. Yeah,
Great.
Exactly.
Thanks.
No, thanks for those questions.
Thank you, Tony. Next in line, Ute.
Yes, hi, my name is Ute Radespiel from the Institute of Zoology in Hanover, Germany. So first of all, thank you very much for the very nice talk, and I really enjoyed it. And I have one question that closely follows up on Anthony's comment in a way, because it's kind of a comment. I would like to hear your opinion about it. When people talk about these early adversities, some of those parameters, for me, first of all, it seems to be very critical or also confusing where you draw the line and say where is early is finished or is it still on, and all these factors that you have been mentioning that go into your score. Some of them have no really an age to it or are early, but they are kind of continuously existing throughout life in a way. and others, it seems so deliberate to me, whether you say 1 year, 4 years, 3 months, what is the scale, or how is it explored or determined where you cut the line? And this would be my first, would like to hear your opinion about how to approach these issues. And the second was, if you would, you said, I think during the talk when you were presenting the results on the heritability, there is so much unexplained variance left, and you said something like, I could talk about it more, but I would like to hear your opinion about what could explain the unexplained.
Yeah, great questions. Okay, so first, when we're thinking about these sources of early adversity, we decided to make really strict cutoffs, all of which occurred before the females in our population reach menarche. And the average age of menarche, the onset of sexual maturation in this population, is about 4.5 years. So the longest source of early adversity that we look at is maternal loss before age 4. All of the other sources are in the first 1 to 2 years, or even at birth, which is large group size at birth. And we decided to make a really explicit cutoff, even though these are, I agree, continuous factors. And so assessing the sensitive or critical periods for these early adversities in this system and in many others, including humans, is an open question that I think could be really fruitful in the, if you have the right data set with the right amount of, data power to assess these types of questions. So yes, I think another related question is, how these early life environments affect survival in the earliest life stages, right? So how much of a selection bias do we have in our adult population? And so I've done a little bit of work on that as well that I, didn't get to talk about today. So yeah, there's all these important questions about how we essentially define these things that I think in the Amboseli system and in many systems who are asking these questions, it boils down to thinking about the biology of the organism that we're talking about, but also thinking really carefully about where these cutoffs are. Okay, and then to answer your second question about the unexplained variants, oops, yes, I stopped my screen share. 1 second, I was going to pull up the graph, but maybe. Okay, so excuse me, even in the heritability model, which is the best model, over 75% of variance in this model was unexplained. And so we have a few hypotheses about why this might be the case. And so, one thing is that there were environmental factors that we weren't including in the model. A second hypothesis is potentially that because age at first grooming is somewhat of an integrated trait, relying on a lot of different physiological and, social learning inputs, as someone had mentioned earlier, and muscle development, as well as social development, there could be a lot of unexplained variance because it is a really complex trait. Some of it could be, as we talked about earlier, the observer effort in terms of, is how well is this first observed grooming pattern really is. And then finally, what I think would be intriguing to measure, maybe it's the most intriguing because I'm interested in this, is the potential for, indirect genetic effects where the genotypes of your social partners affect your genotype. That's been shown in adult grooming behavior in the Amboseli baboons as well as other systems. So understanding how much IgEs play a role in the development of social behavior is an open question. So I don't know what explains this unexplained variance. We have lots of hypotheses that we need to really hash out.
Okay, thank you.
Thank you.
Thank you, Lisa. Next question comes from Clara.
Hi, I'm not back on the multiple screen yet. This is Clara B. Jones, Social Evolution, retired. I just have a point of clarification. Something that always confuses me in the primatology literature is that bond, the word bond, always seems to be assuming that that individual relationship or interaction is a positive interaction. Couldn't the interaction be selfish or coercive or manipulative also?
Yeah, so when we're thinking about social bonds here, we're specifically measuring grooming, which is, an affiliative behavior. But I think you're right in that looking at other types of social interactions, like aggressive behavior, for one, we have aggressive data as a potential mediator could be an interesting and, fruitful avenue of future research. I worked with a different undergrad to ask if age at first aggressive behavior was heritable. And we did find that it was also heritable, and actually much more heritable than age at first grooming. So I think, yeah, we focus a lot on social behavior, maybe because, I don't know, we're humans and we like happy, I like happy things, but, aggressive behavior is also a really important feature of the environment. And lots of people focus on aggressive behavior. I'm not saying that we don't, but yeah, that's a great question, Clara.
Yeah, just quickly to follow, thank you. Very interesting talk, by the way. Just to follow up on your answer, let's see, I'm forgetting what I wanted to say. Oh, yeah. It seems to me that data on who initiates first and in relation to age, sex, and dominance rank might give you some information about the nature of the interaction, whether it's positive or negative.
Yeah, so we have information about whether the interaction, we do have that information, right? Like who's initiating, what their sex is, what their social status is, if they have a defined social status, as well as, the type of behavior. So is it a positive grooming behavior or is it a more aggressive behavior? So we do have that information. I've been focused on the grooming side, but like I said, I think, focusing on the aggressive interaction side could be very fruitful as well.
Thank you.
Yeah, thank you.
Thank you, Clara. And next we have Jinwoo. Oh, sorry, Jinwoo. I should have called you before. But...
Oh, so it's okay. Hello, I'm Jin Yu from I'm a PhD student from University of Strasbourg. I'm working on the personality of bush karoo rat in South Africa. So my question is that one of your conclusions is that social bond increase female survival, and you mentioned that you observe, I think, about 30 deaths of the females. So I wonder, do you know what are the main cause of death of these individuals? Like, is it mostly internal reasons such as old age or disease, or do they mostly die for unpredicted events such as predation?
Yeah, so we had 74 deaths in our data set of 199 females, and it's really difficult to get at what exactly they die from. For some individuals, we can, we recover their carcasses, and so we can infer, that there was a predator attack or it was human mediated. But for others, we don't know, and so I think it's a really interesting question of, exactly what these individuals are dying of. But we don't have the resolution in the data yet to ask those types of questions. Yeah, great question.
Okay, thank you.
Thank you. Suleima.
Hi, Suleima Tang Martinez, retired from the University of Missouri in St. Louis in the United States. And I have 2 questions. I think they're somewhat related, but maybe not. And it's possible I had to step away for a second to deal with my dogs for more than a second, a few minutes. And so it's possible that I missed this. I was wondering why you chose age at first grooming as your starting point for the study, and why, for example, not how much time the males and females, the offspring spent with the mother. Was there something specifically about age at first grooming that you thought was particularly relevant? And then the second question has to do with the differences between the ages at which males and females start grooming, and then how those two lines compare. And I'm wondering if you have any idea what causes the differences. Are you assuming that the differences between males and females are genetic, or could it be something about the differing behavior of the mother towards male and female offspring, whether mothers, for example, are more likely to stay in closer contact with their daughters as opposed to their sons, whether they're more tolerant of daughters to sons, those sorts of questions. So basically, what you think causes the differences. And I thought it was a very interesting talk and fascinating topic of effects of early environment. So thank you very much for presenting.
Thanks, Luana, for those questions and comments. So we decided to assess age at first grooming because it is this really important social developmental milestone. I agree that, we could have also assessed, time spent grooming throughout the juvenile period, and that is an interesting factor as well. There are other people on the project who are working on those types of questions as well. So it was partially a logistical problem for one. In addition, this started as a summer undergraduate project, so we wanted to have something that was discrete and able to have some results in addition to, helping Madison learn R, because that's a big, a big task that she did really great with. So part of it was logistical, but also, we understand how different, sexual milestones differ in the baboons. So, previous work had looked at, age at menarche, age at testicular and rank, development and acquisition. And so we hadn't really focused on age at social milestones. And so that's why we decided to go with age at first grooming. To answer your question about, what differences, like what do I think explains the differences between males and females? I did show some data that the mothers don't seem to differ in their rates of grooming with their young offspring. But I don't know, we haven't asked, how mothers might differ between their proximity to males and females. I think that's a really good question. My hypothesis is that it's probably the social environment and just potentially innate differences between males and females about what unconsciously, but what relationships matter in adulthood. So in this species, like many Sarcopithecine primates and other species like hyenas, we have a maternal or females stay within their groups, right? And they have rank acquisition that's determined by their matriline, right? And so social behavior is really important. Affiliative social behavior is really important for females as they develop. So that's one explanation for why we might expect this difference. Whereas for males, aggressive behavior is more important because they are going to be dispersing, and then they acquire rank based on their competitive ability. And so when we looked at age at first agonism, we actually found the reverse, where males were agonizing, using agonistic behavior earlier than females. So that's some more evidence that suggests it might be just sex differences related to, the biology and life history of the organism. I do think that there's probably some variation that's explained by the mother, but we did look at, a variety of potential maternal effects, and we didn't find really any strong evidence for most of them, although for males, we did find this maternal rank effect. So I think, the mother is also important, but I think really it probably lies in the differences between males and females and what fitness related behaviors are important in adulthood.
Great, thanks. And I guess one follow-up, is there any evidence of different physiological, endocrinological differences in the mothers when they're pregnant with a female versus a male?
Yes. I don't think it, I'm trying to remember. So there was an honor student project looking at thyroid hormone, I want to say, and differences between males and females in rank, and I don't remember their results. But Laurence Desquire, who is a lab manager in the Alberts lab, does a lot of asks a lot of questions about the endocrinological components of these different behaviors and life history strategies, and her work is really fascinating. So if you're interested in that, you know, I suggest getting in touch with her and reading some of her papers there. Yeah, thanks, Zulema.
Yeah, thanks.
Thank you, Zulema. Eduardo and Raghavendra, I'm going to skip us, and I will call Adrienne.
I'm sorry, did you say?
I will skip us because we have a student. So,
okay.
Adrienne, you can ask the question.
Hi, so I'm Adrienne I'm PhD student from the University of Strasbourg in France. I'm not studying sociality or social evolution, but I'm really interested in this. And I will be interested to know if you know, or if you have some insights either from this species or other primates or any other similar species in which this could have been studied, on what could be the link, the mechanistical links between this harshness or these stressful conditions or situations and the variation in their ability to socialize, if they may be modulated by some changes in neurology, like some changes in I would say cognition, or maybe it's being studied from the point of view of at least like hormonal changes or any other physiological state that may modulate all this. Because, for example, I understand that things like loss of the mother, which is directly related to the way that they can interact and learn how to interact, makes a lot of sense, but it is a bit more difficult to understand, I think, a priori, how, for example, periods of drought or seasonality or something like that may affect social behavior in long term. Or how will be the link from that to social behavior, specifically in the long term?
Yeah, that's a great question, Adrienne. So we've looked at the physiological components mostly in terms of glucocorticoid hormone, right? So hormones related to the stress response. And so Stacy Rosenbaum, who I believe is at Michigan right now, when she was a postdoc in Beth Archie's lab at Notre Dame, she assessed these links between early life adversities, adult social bonds, and glucocorticoids. And she similarly found that there really wasn't a mediating effect, but instead more independent effects of adult traits and on or early life adversities as well as adult social bonds. And then I was just going to click through, bear with me. We also did a similar analysis with, here we go, where I assessed glucocorticoids as the mediator, and we found that, you know, this cumulative adversity index increases glucocorticoids by about 10%, and then similarly a 1 unit increase in glucocorticoids affects survival. So decrease in glucocorticoids increases survival. Oh, and I didn't change this. But here there was still this really weak mediation effect. So it suggests that these, you know, we haven't found the really strong mediator linking these effects, or that there are lots of small mediators, right, that all together link early life adversity to survival. And, you know, it's a lifelong process. So I kind of feel like it's probably a bunch of small things that link these processes together. a little bit tangential to your question, Emily Levy worked on the effects of early adversity on immune response, right? So how it affects immune response. And I believe she didn't find really strong evidence that many sources of adversity affected immune response, but I believe that she's working on writing that up now. So stay tuned to that because it's really fascinating work. So yeah, we're trying to get at the, underpinnings, right? And then some folks in the Tung lab, so like Jordan Anderson, is looking at some sources of early adversity and how they might affect DNA methylation. So I think one really powerful thing about the Amboseli Baboon Research Project is there's just so much data, which is really powerful and great. But there's so many wonderful people working and have interest in all these different levels of analysis, right? Which makes it a really powerful system. Yeah.
Yeah, thank you. That was nice. If I can just ask a little question in that line, do you know if there is anything on how this kind of disruption on their sociality or their interactions can affect aging or like the late life?
Yeah, so Jordan Anderson also looked at aging effects, and Chelsea Weibel, who just finished her PhD, has some stuff coming out too. So I don't remember exactly what the results are, so I'm hesitant to say anything, but there does seem to be effects of these social behaviors on DNA methylation and aging the biological clock too. Yeah, great question.
Thank you very much.
Thank you, Adrian. Okay, Eduardo, you're next.
Yes, thanks. Thank you, Liz. We're few now. We're most of us, we know each other well. I am pondering what to ask. And for those of you who may not be aware, I'm meeting with Liz tomorrow in the morning, and she's agreed to join my graduate course where we're reading her work. So what I'm asking is not so much asking Liz. I mean, Liz is almost the messenger of a project we all look up to. It's the kind of holy grail. I mean, we all wish we had the 52 years of history and the data and the resources. So that's because, I mean, this is more for Susan and Jenny, and that's because we're going to keep raising the bar because they have shown us that they can react to it. So some of the comments, Liz, I'm not even sure if they're for you, but I was left with many questions, but it isn't specific. I love your talk. We're going to talk some more about it. I wonder if because of because the strong position where they are, my reaction is, well, isn't this the time to start checking from some other angles if the story holds? So for one thing, it is misleading. I think it is misleading to keep talking about social bonds when they mean grooming. So a very concrete question is, why call it social bond if you know it's only grooming? That's basic. And I know the answer, because to get a paper out in Science or PNAS, etc., and connected to humans, social bonding is going to go better than grooming. But regardless of that, I mean, wouldn't it be amazing, instead of collecting more data, collect fewer data, much more precise? I mean, we've had questions. Adrian was just asking mechanisms. I mean, Gene Oldman was 40 years ago looking at foraging, not drought. Drought is the kind of coarse proxy that those of us without resources use. We go to the weather station and we just get rainfall. It doesn't tell us what the animal is suffering. So why not challenge? I mean, it'd be amazing because they can do it. Challenge these pillars on which we keep getting data that, I mean, are those analyzing the data, collecting the data? Are they watching the baboons? Do we have people still who are going to be analyzing the data, going to the field and actually watching the baboons and getting to know them? So getting to the mechanisms, because that's what can be done, as opposed to this big data approach, which is a causal salad of, I throw there a drought and an early life adversity, and nobody knows anymore what it means. So it's fascinating. I wish I could be discussing. I mean, we've talked about this with Susan, and I'd love to discuss it some more, because they can do it. And even if it slows down for a while, they'll keep showing us the way. Glucocorticoids, maybe, well, I mean, yeah, it could be mediator, but when they started doing fecal samples 30 years ago, we were thinking about cortisol as stress. Now we know that we don't even know what fecal glucocorticoids, which in many systems people are not measuring anymore, period, because we don't even know what it reflects that's happening in the brain. Can be insulin, can be any number of hormones. So, but revisit the social index. Let's get something else besides grooming to see if the story holds.
Yeah, so I mean, I think there's, two competing interests, right? Like that you brought up. There's the, overarching, you're doing this long-term study, and there's definitely positives, right? But there's definitely negatives in terms of like the granularity of the data, right? I'll say that I somewhat disagree about completely stopping collecting all this data to get more fine scale data, because we have often gone back and reanalyzed, with more data and seen that the story changes. And so I think by continuously collecting the same types of data is a really powerful approach to assess how patterns might change with time that we don't have in a lot of systems. However, I will say that, when you think about the glucocorticoid data that you brought up, I agree with you, but also, 20 years from now, we might have further insights that we say, oh, I wish we wouldn't have stopped collecting the glucocorticoid data, or maybe we wish we did. It's hard to predict. I think one of the really strong positive things out of the Amboseli project is that they've been collecting these types of data for so long, and it's somewhat forward thinking. When there's new, types of data like, genetic data, we started collecting it somewhat early, and then now we have this wealth of data that we can use for things like pedigree type analyses. But I presented data using the long-term data set, and many postdocs and students and researchers do that. But there are also people who are going over to the project and collecting this really fine scale data. So one of the graduate students in Susan Alvarez's lab right now, Maria Crichton, is actually testing hypotheses about foraging effects on social behavior by looking at, fine scale measurements of what the baboons are actually eating. And so, that's somewhat limited to the time span of a PhD or a postdoc. For me, I started my postdoc in 2020, so no one was going to anywhere, unfortunately, although that's a little bit of a cop-out, but that is what it is. I got lucky because I could work with the long-term data instead of being able to collect fine scale data. But I think that you're right, that combining the long scale data with the fine scale data makes a really powerful approach.
And again, this is, we should have, it'd be so amazing to have an opportunity, to talk for hours. It's amazing. I haven't seen the baboons. I haven't been to Amboseli. But my goodness, we've all, I'm discussing these papers tomorrow in my course, right? What better measure that I do believe they're doing wonderful, amazing things.
Yeah, but they're like all data types, right? Even if you're doing experiments or you're doing more fine scale observation data collection, there are, limitations, right? And the limitations are just different depending on what type of data they are. And
right, but where I think I see a difference that I did not, if I conveyed a sense of there's some negative aspects, I don't even want to call it. I don't see anything negative. I think of it as missed opportunities more than negative. So I see here missed. I wonder if, but of course you need to stop doing, time is limited. So you need to stop doing this so that you can do that. But it's a very simple question. Why not reassess all this in light? Social bonds, one of the participants was asking, I think it was Clara. If we're going to connect it to humans, there's an awful lot to be said about what a social bond is. Why not, for a year, collect something different, speaking of a bond, and then compare it to what grooming is saying, right? In a very much more, and maybe they're doing it, but sometimes a better answer may come from a shorter, more thorough study. than a huge one. I mean, that's how we proceed in medicine, right? Sometimes we just need that very well-defined clinical trial with a small set of people, all of the same height or whatever, and only then we seek external validation. But internal validity is what worries me a little bit about these indices and this tabulation of droughts, that going for the external validity, we may be risking the internal validity of the measurements.
Yeah, I mean, I agree with that. I think that we benefit by doing both, right? We do this long-term data collection, and we also have more fine scale pinpoints. And then to your point about social bonds, I mean, part of the reason we call these grooming relationships social bonds is because it's a science communication thing, right? Like, people understand social bonds. Only a certain subset of people really understand what grooming is, right? But you're right, we lose some specificity when we think about more general terms, right? It's like modeling trade-offs between having a super specific model and having a more general model that might be more applicable to lots of different systems. In terms of like other measures of social relationships, we do have people working on, different ways to measure sociality, including thinking about proximity, thinking about more larger scale social network analyses using grooming, but also using things like aggression. So there are things that will be coming out of the lab doing these types of things. But it is essentially, it is a trade-off, and that's above my pay scale as to where we are focusing our efforts. I mean,
you've done terrific. That's something else that I think we're contributing with fine in a friendly audience. We just want to learn. I mean, my goodness, you're going to be. This is better than going for a job interview. You better have these questions here, and you've done great, Liz, answering what you can answer, right? I mean, so fantastic. Thank you so much.
No, thank you, Eduardo.
Okay. Recommend, I'm gonna let you go first. You've been waiting long in the list.
You go ahead. I'm going to talk about something very different. So you go ahead.
Okay, well, let's go back to I go ahead. Thanks, Liz, for a great talk, and also thank you, Suleima, because you pitched on something I wanted to ask. And now that we're clear on the differences between males and females, I want to dig a little bit on that and ask you a little bit more about the differences on the development of this first age of grooming in terms of like, okay, so males do differ in their morphological or maybe the propensity to groom, or is there anything that sets them on a different track from the very beginning? And then the second question goes into, once they establish this first age or first grooming period or say milestone, do they do it with a different type of individual? Like, do females bond first with their mother, or males do it with a different type of individual? And will that have any consequence in their fitness later on?
Great questions. Thank you, Anna. Okay, so in terms of your first question about males and females, we do see that, in adulthood and, early in life, that males are grooming at a lower rate compared to females, right? So the first grooming is at about, 10 to 12 months. So right about here, we don't see any differences. And this is just for the mother offspring grooming, but we don't see any differences until, maybe 7 to 10 months after age of first grooming. But in adulthood as well, we see that females tend to groom more often than males. Males are tending to do other behaviors. So, I mean, I think that's one explanation for why we see these differences between males and females, right? Is that females are just doing it more. We don't know. So, I mean, Madison and I assess the, who they groomed first, right? Oh, this is way back. So maybe, the first grooming partner, but we didn't look, so that's here, but we didn't look at, subsequent grooming partners. And I think that's an interesting thing. Like, are they always grooming their mother before the first few times, or are they always grooming, whatever grooming partner they decided to groom first? Or, once they groom, are they, oh, right now I'm a groomer and I'm going to just participate in this social behavior? That's an interesting question that I think would be cool to assess. Yeah. And it gets at, like this development of this really important behavior that I think we need to understand more about. Did I miss any of your questions?
No, I don't think so. Well, we haven't really looked further, so we don't know if that will have a difference in the fitness.
Yeah, exactly. That's one thing I'd
exactly.
like to do is look at, tying this behavior to fitness. Yeah.
Cool. Okay, thank you. Raghavendra?
Yeah, I am Raghavendra Gadadkar from the Indian Institute of Science. I sort of delayed asking my question because I don't have a question. I have a comment. I want to say I really enjoyed your talk very much. I was amazed at the magnitude of the data, but even more about the depth and precision of your analysis. So it's absolutely wonderful that I sat up late in the night. It's 11 p.m. for me, but I really enjoyed it. But I must confess that there's another reason why I enjoyed your talk very much, because as you were speaking, I was feeling that there's uncanny similarity between what we find in an insect model and your baboons. So we work on social wasps, and the workers feed the larvae, and there's a lot of natural variation in how many times a larva is fed. And we have shown that the well-fed larvae go on to develop into adults who seem to feel more hungry. They eat more. They become more likely to become reproductive, live longer, have higher fitness. And conversely, the larvae which are fed less for whatever reason develop into adults who don't seem to feel hungry. They don't eat enough, even if food is available. They are more likely to become non-reproductive, have lower fitness, have lower lifespans. As if that's not enough, these early life problems of some wasps can actually be reversed in the adult stage. Because when my students hand feed the wasps in the adult stage, then they are more likely and increase their rate of food intake over and above what they would do on their own. Then they're more likely to become reproductive, higher fitness, and have a longer lifespan. So it's uncanny that an insect system and a baboon system, at least at a superficial level, are really, really very similar. Of course, we don't know what the mechanisms are, but that's what sort of kept me absolutely fascinated throughout your talk. So thank you very much.
Well, thank you. I mean, and thank you for staying up so late to the talk. I don't know if I'd be able to do that. That's way past my bedtime. Yeah, so I agree. I think that, I studied baboons my postdoc. I study fish now. There's a lot of parallels in many different systems, right? In terms of thinking about these general patterns. And so I'm currently working on a meta-analysis and review type paper asking about these general patterns that we might see. So hopefully that'll be out in the next year or so. So stay tuned to that. There's a little plug for that paper.
Great. Thank you very much, Raghavendra. And I think we don't have questions on the YouTube, right, Lauren? No questions. Okay, good. So thank you. So I think with that, I would like to thank again Liz for a great talk, for staying up with us with the discussion. That was awesome. And I'll invite you all to come to our next seminar next week. And with that, I'm